Hello everyone, in this video we are going to discuss about prokaryotic gene regulation, that is switching on and switching off the gene expression using the lac operon, which is a classic example of a genetic response or gene response to an environmental change. Here we see how the lac operon is regulated at different contexts, such as in the presence of lactose, in the absence of glucose, and in the presence of glucose and lactose, etc. How does these metabolic substrates such as glucose and lactose are manipulating the lac operon? Before we shall see what is a operon. Operon is a region or sequence that is located between the promoter and the structural genes, which binds to the repressor protein and switches off the gene expression. Lac operon is a polycystonic gene. What does it mean? Polycystonic genes means group of genes are under the control of same promoter and operator. Let's see what are the genes required for lactose metabolism. LAC-Z, which encodes permease, LAC-Y, which encodes beta-galactosidase, which breaks the lactose which entered into the cell into glucose and galactose, LAC-A, which is a trans-acetylase. The operator region, when during the switched off condition, it is bound to the repressor and it doesn't allow the RNA polymerase bound to the promoter region to move forward, and it is always repressing the gene expression or inactivating the gene to express. The repressor is coded by the LAC-I region, which is upstream to the lac promoter region. And also here there is another region, cap site. Cap site is a positive regulator, meaning it enhances the transcription, whereas the repressor protein encoded by LAC-I region upstream to the cap site and lac promoter, by binding to the operator, it will inhibit the RNA polymerase and inhibits the RNA transcription of LAC-Z, Y, A genes. So here we have 4 different types of conditions. In the first condition, there is low glucose and lactose available, meaning the cell is deprived of energy because of shortage of the glucose and it is looking for the energy source, and it tries to utilize the lactose available by switching on the lac genes, which helps to break down the lactose into glucose and galactose. So in this condition, as lactose is available, it converts into the allolactose and binds to the repressor protein and promotes it to dissociate from the operator, and it clears the operator region. So the RNA polymerase bound to the promoter region of the lac operon can easily move forward and encode the genes Z, Y and A required for lactose metabolism. In the second situation, where there is high glucose and lactose is totally absent, here when there is no lactose, there is nobody to remove the repressor from the operator. So the repressor is bound to the operator side, blocking the transcription of the LAC-Z, Y and A. Low glucose, lactose unavailable. In this condition, there is very low glucose and lactose is completely unavailable in the environment the cell is surrounded with. As there is no lactose, so no allolactose to remove the repressor from the operator, so the operon is switched off in condition. And in last condition, there is high glucose and lactose is also available. So in this condition, as there is lactose available, it converts into allolactose and it removes the repressor protein from the operator. So it promotes the gene expression in this case, but the expression is very low or basal level of expression. And what is binding to the cap site? How it is positively switching on the lac operon. The cap site, by binding to the CAMP molecule, which is a signal molecule indicating energy shortage in the cell, it will bind to the cap protein, and this cap complex formed by combining with CAMP binding to the CAP site will positively enhances the expression of the LAC gene in the presence of lactose in the surrounding environment. CAMP indicates that there is shortage of energy, and the CAMP, by combining with CAP in the presence of lactose, it binds to the CAP site and it tries to enhance the gene expression of the LAC operon in order to utilize the lactose available in the environment.